Okay, so let's talk about some advantages to being multicellular. To understand the big picture, let's just compare you as a human with all your cells to a unicellular thing like a protist or even a bacteria. If your entire body is one cell, then when it gets damaged or hurt, the entire organism is dead. You, on the other hand, when you get a cut on your skin and you damage your skin, you can go through mitosis, as we've discussed before, to repair and grow new skin. Because you have so many cells, you actually can assign them different roles. Our poor protist and bacteria friends have to have that one cell do all of the body functions. That includes anything from grabbing energy to movement. Everything is performed by one cell. You have cells that can do different jobs. They are very specialized to do their own job, so no one cell is bogged down with a bunch of tasks. This is referred to as cell specialization. It's a sign of a certain role or a certain job to the cell. So how do you get to this point where you have all these jobs and all these roles for these different cells? Well, first of all, you start out with something called stem cells. Let's assume in this analogy, a stem cell is a pile of sand from the beach. You just throw a pile of sand down. It's not in any shape. It's not really doing anything. It's just a pile of sand. You'll remember from other units that there are, 6-ish feet of DNA per cell, and it's all the same. So your brain cell, your kidney cell, your liver cell, they all have that 6 feet of DNA. Now imagine this 6 feet of DNA has a bunch of light switches on it. Each of these light switches can be flipped on or off depending on what we need the cell to do. Stem cells, they're pretty much all turned off. Each light switch affects one section of the DNA. This is called a gene. That means this gene can be turned on or off to do different things. There are a lot of things that can turn the switch on and off. One of the big ones that you'll know about is hormones. They basically act as a light switch gene trigger. When they flip it on, it reads that section of DNA and it creates that specific protein. Some cells are going to require a lot of light switches to be turned on, some are going to require very few. It all depends on the complexity of the cell. So stem cells, by being activated by hormones or some other chemical messenger, are going to start flipping on light switches and becoming a very specialized cell. They'll decide that they're going to be a brain cell and they'll go along that path and become a neuron, or they'll decide they need to be in the kidney and they'll go along that path and become a nephron. This is called cell differentiation. So the big focus of this video is going to be on two very specifically specialized cells. These cells are called sex cells or gametes. This is the sperm and the egg. These are the main reproductive cells in humans, and they're called sometimes different things in different eukaryotes, but they're created by the same process of meiosis. Meiosis is very, very similar to mitosis. Remember mitosis here? Prophase, metaphase, anaphase, telophase, cytokinesis. And then we go into interphase and repeat it as necessary. Please make a terrific cookie. Meiosis is a little bit different because, as you know, a sperm and an egg are going to combine to form the first cell of a new organism. When the sperm and egg combine, they form a zygote. A zygote has the same amount of cells as the rest of your body. Sperm and egg are not body cells. Body cells, or somatic cells like skin, kidney, liver, brain, are going to have 23 pairs of chromosomes. This is also called a diploid cell. So they have 46 total chromosomes. We want the baby that's created from the sperm and egg to have the same 46 chromosomes, so we can't combine 46 and 46. That'll give us 92. That's not what we want. So the meiosis process has to create two specialized cells, sperm and egg, that have half of the DNA. This is called a haploid. They're only going to have 23 chromosomes total, not 23 pairs. They're half of 46, which is 23 total chromosomes. So here's the process of meiosis. So we're going to look at the big picture of meiosis for a quick second before we add in all of the extra color. And we're going to use it orange just so you remember it's not the same as mitosis, but it is very, very similar. Please make a terrific cookie still applies. We still got prophase, metaphase, anaphase, telophase, cytokinesis, but now we have two of them. We have prophase 1, 2, metaphase 1, 2, anaphase 1, 2, telophase 1 and 2, and cytokinesis 1 and 2. And they all do the same basic things. They still have our prep metal apart 2 cut. The difference in meiosis, besides having a second splitting stage, is going to happen in prophase. And the thing that happens in prophase 1 is going to affect all of the stages from here on out. So everything above that purple line is meiosis 1, everything below that purple line is meiosis 2. Alright, so looking at prophase 1, we still go from chromatin, a bunch of noodles, then we start to condense into the X's. However, if you'll see, the X's are kind of overlapped, and that is on purpose. Since the specialized cells we're trying to create are sperm and egg, we do not want to create identical copies of the original cell. The reason for this is because we know that diversity increases your chance for survival. So if we're creating sexual reproduction cells, we want them all to be different. We want them all to produce different offspring with different combination of traits. So how do we take the mitosis steps, which created a perfect copy, and change them a little bit so that we now create different cells at the end of the process? The process is called crossing over, and it only occurs in prophase 1. So assume these are our chromosomes floating around in prophase 1. You got a red one and a blue one, which means they have different traits. Mitosis would create 2 reds and 2 blues, so that when we split, each cell ends up with the red and the blue, and we end up exactly like this. Meiosis, however, does not want this. The X's are actually going to overlap. And where they overlap, they are actually going to smudge or kind of exchange some of their DNA onto the other so that they can crisscross some of the blue and the red. So after crossing over occurs in prophase 1 and we move into metaphase 1, the X's now look something like this. They have now exchanged a little bit of the red and the blue in certain sections. It's not always the same section, it's not always the same leg, but they are going to blend a little bit of their DNA. This will become much more apparent why it's going to create completely different cells as we go through the process. So let's go back to our main picture. So we will assume that we got 2 blacks and 2 greens. They have crossed over and we can move into metaphase. So we have had our crossing over occur. We move over into metaphase and you'll see we have a black X on the top with a little bit of smudged or exchanged genetic material from one of the green X's on the top right, another X with a little bit of black on the top left, another black with green on top right, and green with black on top left. So we basically have 2 identical smudges of each type. So now we move on to anaphase. Anaphase pulls them apart, we get half of each X, and we stick them into their separate nuclei as it reforms in telophase 1. So now, after cytokinesis 1, we have 2 different cells. They have 2 X's in each, just like a normal skin cell would. However, at this point, we are not going to go into another synthesis phase. We are not going to double those. We are going straight into meiosis 2. This is the difference between mitosis and meiosis. Meiosis 2 does not replicate through synthesis again. So we are going to go straight into another splitting phase of prophase 2. Prophase 2 starts to dissolve the nuclei again, spindle fibers and all that form. It's prepping to divide again. Metaphase lines them all up in the middle, getting ready to pull them apart, and then we pull them apart in anaphase. So we've pulled half of each X, half of each chromosome into a chromatid, and pull them apart because we're going to go ahead and split it again in telophase 2 and start to reform the nucleus. So now that we pulled half of that apart, cytokinesis 2 has 4 genetically different sex cells. We got top left, we have a full black with all the genetic information from a black chromosome. Top right, we get some black on the top with some green on the bottom. Bottom right, we got all green. And bottom left, we got green on top with black on bottom. Each spot on this X is a different trait. So just because one may have half black, half green, the location matters as well. And remember, colors are just rudimentary explanations of it. You could have a multitude of different colors because you have so many different traits going on. This is just a simplified version, but we still end up with the same result. 4 genetically different haploids because they have half the DNA. So let's count up our chromosomes. At the beginning of prophase 1, that is the start of basically our splitting phase. So we've already gone through our G1 and S and G2. So we have double DNA. Remember, the normal somatic cell, the normal body cell, has 23 pairs, so 46. Since we've already doubled, prophase 1 has 92. We got 92 chromosomes in there. And we're going to continue to pull it apart and split until we can get to our first cytokinesis and pop them apart. So at the end of cytokinesis, we have 46 in each one. The 46 chromosomes is our 23 pairs, so we've hit the somatic cell. So if we are doing mitosis, remember, this is where we stop. We have the perfect copy of the original cell that we wanted. We do not go into another replication phase for our DNA, but we need to get half of this. Remember, sperm, egg can't combine 46 and 46. That will not give us the correct 23 pairs of chromosomes. So we go straight into our other split to get these sex cells. And by the time we get to cytokinesis 2, we have split them all into 4 cells with 23 chromosomes each. This could be 4 sperm, this could be 4 egg, and they're combined with 4 more sperm and combine with 4 more egg to make our 46. 46 is the full blueprint, so now we can build the baby from the zygote, which is 1 sperm, 1 egg. And just because we can't see the chromosomes underneath, we're going to color them real quick so you remember that these are 4 different cells. Each one of these sperm combining with an egg would create a different combination of traits. This is why you and your sibling, even though you have the same mother and father, do not look identical. Identical twins is kind of a mistake in the zygote where it splits and it pops apart, and then they both continue to multiply into a baby. Fraternal twins is when 2 eggs get fertilized, so they're still going to look very different. You even have fraternal twins that are different genders. And as far as evolution thinks, let's say that the pink cell over here has really good traits and the orange, blue, and green do not. If 3 of those do not survive, but at least the pink one survives, then that species can send on DNA and procreate and continue to survive. So nature counts that as a win. So the diversification of its offspring and the diversity of the population make it stronger and make it more stable. We went through all of this process just to get 4 different haploids to strengthen our gene pool.